Hello students, welcome back once again with the human histology videos. Myself Professor Dr. SRHU Dubey, Professor in the Department of Anatomy, Himalayan Institute of Medical Sciences, Swami Rama Himalayan University, Dehradun, Uttarakhand. In this lecture series, I have introduced you the various concepts of the human histology, and in that series today we are going to discuss some of the neuronal histology which I present. Neuronal histology is very complex histology and vast in itself. A series of the lectures could be taken on this histology, but I am going to touch only the basic ones which may require for the undergrads medical students. This is the nervous tissue, and this nervous tissue is of various organs. I hope you can guess this organ. Can you guess this one? A butterfly structure with central empty canal sort of structure. Yes, you are right. It is spinal cord, and this spinal cord structure looks like central gray matter, which is always a butterfly shaped structure, and outside the white matter is present. In this histological slide, we see the dark pink structure is actually a gray matter. Central canal is there, and then outside this lighter blue structures are basically white matter is present. In this gray matter, we see this clear-cut dark triangular shaped pinkish blue dots, and these are the basically neuronal structures of the cell body structure, right? So, they appear triangular in this area, and then in between these two there are the multiple dendrites are present, or the network of dendrites are present here and there. Also, if you see them, they are the clear cut round structures are present which are empty and white colors here. Can you depict it? I am putting my cursor here, this one. These are basically blood vessels, and this area is not devoid of blood. Neuronal structures are very sensitive to the oxygen. So, a rich blood supply is also present here, but in the form of very small vessels. So, whenever we take a section, these small vessels are also present here, but in the form of empty structures. This is the central canal where the CSF is present there, and then outside we see various groups of the neurons, and in the neurons what we see, this is the axonal part of the neurons. So, its axonal part is basically vertical here, okay. So, these vertically coming groups of axons are present here, which that's why we see it is a scanty structure. We already know these all are covered with the fat layer is there, and that's why it is there as white matter, okay. So, axons are present over here. Any disease of this area may cause the damage of the axon. In the histology, we see this is the areas where white color gaps are present, and that gives to the basically axonal structure. Now, we switch to this diagram here. It is a slide of a cerebral cortex. What we see here is a ground pink color structure, slightly lighter here and darker here. Then we see dark pink, slightly bluish tint in it. Various cells are present in this structure. What we see, it is scanty here and short in structure as well. Here the cells are a little bit bigger as well, and they are more in number. Now we see, can you see this tail-like structure? Can you see this tail-like structure? This one, basically it is the cell body, and this one is the area of axon where the axon have just started. And since it looks like a triangular, it is named as pyramidal cells. So, many pyramidal cells are visible here. Basically, this is the part of the neuronal cell body. So, we see many layers of the pyramidal cells are present into the cerebral cortex. In H&E staining, it is not possible clearly always to differentiate into the cerebral cortex in the various layers. Also, it also depends from where you have taken the tissue for to depict it, so that you can depict the various layers. In the brain, you know that changes in layers in cerebral cortex and parietal, it is different, but if you take it from hippocampus, it is different. In between, there is also changes here. So, at what place you have taken it, that can help you in understanding there. And if you still observe 1 layer, 2 layer, 3 layer, 4 layer. So, this sort of layers are visible into the cerebral cortex. But again, I am telling you, this is not so clear for the beginners. It's a little bit difficult to understand the histology of cerebral cortex, right? But there is a good news as well into the another organ of the brain, and that is the cerebellum. Here into the cerebellum, it is one of the most interesting and the histologically beautiful organ. Why I am telling you? Because if you see this gray and white matter in this, at the junction, you see this special type of cells. These are the flower-shaped cells, and these are named as Purkinje cells. So, into the cerebellum, we see a layer of the Purkinje cell, which can go easily identifiable Purkinje cell layer, clearly arranged in a layer very clearly and divided into the structure into the one lighter zone, another this dark shaped zone. At the junction they are present, and that is undoubtedly the slide of cerebellum. So, in cerebellum, Purkinje cells are clearly visible. Then you also see other structures that the neurons are present, the pyramidal cells are present. Of course, see another sort of neurons are present, which is understanding and analyzing this is beyond the scope of this video, and then you can understand this. So, these are the 3 basic slides of the spinal cord, cerebral cortex and cerebellum. Beside this, there are the another slides of ganglion, peripheral nerve structures also there, which undergrad should understand them. But these slides are not very well visible under the HND staining. We need spatial staining. If you do certain spatial staining, that these slides can show their real magic and the what exactly present in histology. Why it is so? We know that there is a myelination is present over the nerves, and the myelin is made up of fat dominating chemicals. This fat dominating is hydrophobic, and our dyes HNDEs are basically water based dyes. So, our dyes, when we are staining the nervous tissue, are unable to go deep into the nervous tissue and make it more differentiable. Rather, we see almost many structures are samely visible. That is why under the HND, it is not advisable to go into the depth of brain histology. Rather, go into for the spatial staining, and if you do the spatial staining, you can see various layers into the histology of nervous tissue. So, with this basic ideation of the nervous tissue, I thank you all once again for understanding this topic and patience listening. Thank you.